In prokaryotic replication, there is only one origin of replication that occurs in a bidirectional manner, meaning the same thing that is happening in one direction is happening in the opposite simultaneously, beginning at the origin of replication. The first step in replication is the introduction of the enzyme helicase, which separates the two parent DNA strands so they are accessible to other proteins and enzymes used for replication. The helicase unzips the strands, supercoiling occurs ahead of the fork and increases the coiling tension of the double helix. To relieve this torsion, the enzyme gyrase attaches to the DNA and breaks and reforms the phosphodiester bonds in the DNA backbone to basically untwist the double helix until it is back in its relaxed state. Single-stranded binding proteins are constantly binding to the strands of DNA that have been separated from one another to prevent the nucleotides from forming hydrogen bonds together and returning to a double helix shape. Once the replication bubble is formed, RNA primase will attach to the parent DNA strands and lay down an RNA primer in a 3' to 5' direction, giving the DNA polymerase a free 3' OH end for the newly synthesized DNA to bind to, by the formation of a phosphodiester bond to the 5' phosphate of the new nucleotide. Now that DNA polymerase III can bind to the RNA primer and begin to add nucleotides, synthesis of the daughter strand has begun in the 5' to 3' direction. The leading strand is the strand where DNA replication can occur beginning at the origin of replication and continuously add nucleotides to the 3' end without interruption. The lagging strand is where the DNA is still being replicated in a 5' to 3' direction. However, the addition of nucleotides is moving towards the origin of replication in each of the small fragments of DNA it is synthesized in. These fragments are called Okazaki fragments and are the result of discontinuous replication as the RNA primase awaits the unwinding of DNA strands at the replication fork. The DNA is replicated, there are still RNA primers present in the daughter strands, so DNA polymerase acts as a proofreader and removes all these small primers and replaces them with DNA fragments. After the DNA polymerase I activity, there are still nicks present in the daughter strand where a sugar-phosphate bond is missing between the two pieces of new DNA. This is where the enzyme ligase attaches to the daughter strand and seals all of these nicks by creating that sugar-phosphate bond. And that is prokaryotic replication.